Hi, I'm Cindy.
My name is Ruben Narayan.
Hi, my name is Divyan Srivastava.
Hi, my name is Danu Senthilkumaran.
My name is Simran Kaur.
My name is Ankita Rajesh.
My name is Tanisha Rajpur.
Elisa Lewandowski and Bianca Pell.
I'm here along with my partners, Odudu Omondi and Sky Zhang.
I'm Eva Bennett.
Mantra Rajkumar.
Hi, I'm Alana.
Hi, I'm Olivia.
Hi, I'm Carly.
Hi, my name is Lillian Buckley.
I'm Brian Gu.
I'm Neil Abraham.
My name is Freya Fardeen.
My name is Prachi, and our science fair project this year is titled Environmental Effects on Long-Term Memory Consolidation in Drosophila melanogaster.
My project is called Phytodrugs, a novel approach on a planaria model of multiple sclerosis.
And I'm here to present how I developed an innovative alternative model for teratogenicity testing.
My project is on mathematical modeling to determine underlying variability in CDC COVID-19 guidelines adherence.
And our project was about testing the effectiveness of refilling third-party printer ink cartridges and testing the quality of their printing. My project is the effect of EDTA on calcium deposits in arteries.
Our improvised testable question was, what is the effect of environmental lighting on memory consolidation in Drosophila?
Our testable question is, what is the effect of the type of food additive added to fully fermented sourdough bread on its taste and texture?
In this research project, I aim to understand how various factors affect following the CDC guidelines to help protect oneself and others from COVID-19.
The main goal of this project is to help create a wearable device to aid the blind and help them better navigate their surroundings in both a cost-efficient and effective manner.
And our project is the effect of acetaminophen on the survival and heart rate of Daphne magna.
How accurate is astrology, but why do we assign ourselves to these descriptions?
So I was wondering, can machine learning algorithms help predict where the next virus and/or pandemic will come from?
Both of us have friends and family with allergies, and we thought it would be a good idea to research something that could potentially affect the people around us. We asked ourselves, what would be the difference in accuracy with a mass detector between people of color and white people? Our hypothesis was that the detector would be more accurate for white people than people of color because of the inherent prejudice in AI.
So our curiosity took over, and we wanted to put these different products to the test and see how well they actually work.
I wanted my device to be able to take a picture of a urinalysis test strip, then use image processing and color comparison to produce binary data.
Following that train of thought, I wanted to create a packaging that would, instead of detecting enzymes, would detect pH. So that leads me to my testable question. How effective are pH indicators at detecting food spoilage?
This project is divided into two phases. For phase 1, the performance criteria are to build a program to reduce off-target DNA cleavage.
Now the way this works is as the sun moves across its path in the sky, it'll hit these 4 tiny solar panels which are mounted on prongs behind the shield.
After about 20 days of data collection, we saw statistically significant increases in pH in trials treated with seaweed and calcium carbonate.
Before the algorithms can be trained, we must prevent overfitting, or when imperfect data causes an algorithm to be trained poorly.
My initial image capture design was pretty simple. All the camera could do was display a continuous video stream on its web server.
We had an entrance slit on the slide that you can see on the left, and we had a flashlight for our light source.
Neither performed well. It was hard to run in the slime after multiple runs, and some of the slime broke out of their plastic bags.
The performance criteria, or the goal for this device, is to first use the diffraction of polychromatic light to determine the absorbance spectrum of the newborn baby's hemoglobin. To do this, the sensor should first determine the absorption of the finger.
An image is passed through the phone's camera, and then an algorithm that I made, similarly structured to YOLO, predicts what object is inside the image.
Then we took advantage of oxygen's paramagnetic properties and used a magnet to lure the purified oxygen to the second chamber, whilst leaving the carbon monoxide and the carbon dioxide, both of which were diamagnetic, in the first chamber.
We took an initial heart rate, which ranged from 320 to 368 beats per minute, and then recorded the heart rate of 1 Daphnia per petri dish and the number alive in each petri dish every 2 hours over the course of a 12-hour period.
So our conclusion was that the EOS in the fridge was the best overall, and the peppermint birds from temperature was the worst overall.
For the dry paper towel tests, all 3 tests had positive results, which shows an allergen was present.
In my experiment, it was found that EDTA did in fact cause the eggshells to become thinner, softer, and easier to break. The calcium from the eggshells dissolved because the molecule from the EDTA freed its 2 hydrogen atoms.
Genetically modified proteins is often substantially larger than organic proteins.
We scaled up the size of the generator in order to 1. Increase the number of modules in the series for better wattage, and 2. Increase the volume of the ice water container so it can last longer.
Testing could not be done on humans, it was instead simulated using a plastic tube.
And there's also a lot of potential for repairing AFM with food and other amino acid sources due to my data and previous data from studies before.
This knowledge can be applied to other memory-related trials involving fruit flies in humans.
Later on, we hope to carry out the experiment with the actual value that would be considered a mild traumatic brain injury while still maintaining or improving the level of accuracy.
So my conclusion is that fruit peels were more effective than xylem filters, and my hypothesis was correct. I do, however, need more clarity on my xylem filters.
The temperature didn't even reach over 10 degrees Celsius for the full 45 minutes, which was above expectations.
Although we couldn't make a filter portable, a next step we're going to take is to make our filter portable and able to be attached to water bottles so we can filter out microplastics. Thank you.